Hi everyone. I realized that in the last lecture I was maybe a little fumbly. It's pretty early and I'm just still trying to wake up. So welcome to our fifth mini lecture. I wanted to start off by saying that some of the images that we see or that I have in these lectures for the brain are going to have more anatomical things labeled than what we're actually going to talk about. So I'm going to actually go back to the medulla oblongata slide for a second here and give some examples of that and show maybe a little bit more clearly what we should be focusing on or thinking about. Okay, so here is a cross-section of the medulla oblongata. This is the last thing that we looked at in the last little mini lecture. There are a lot of things going on here, okay? This looks quite a bit different from what we saw on the last slide or the slide prior to this where we were looking at the external anatomy. Now we've cut this open and we're looking at the internal anatomy, and I know that there are a lot of things labeled. So let's sort of just walk through. We actually did talk about quite a bit of what's on here. So we did mention externally these two, right? So these would have been the gracile and the cunate fasciculi, rather. So internally, these red spaces or peachy salmony spaces, those are the internal structures of those two fasciculi. So these form ridges on the outside, they form nuclei on the inside, and they are going to be located back here on the posterior wall. So we can start to correlate the external anatomy that we talked about with the internal anatomy. Here I talked about the reticular formation, and so we can see that internal structure. This was not on the external part of the medulla oblongata, so we don't see it there. The medial lemniscus I talked about, so that's going to be decussation of our sensory fibers, so they're crossing over in this area. We have not talked about nerves very much yet, so you can sort of just ignore these. The corticospinal tract, those are our pyramids. So externally, those are our baseball bats on the anterior side of the medulla oblongata. Internally, this is just that region that they take up. Olives on either side, again, looking at the internal structure. And then inside of that olive, we have sort of a specific region that forms the inferior olivary nucleus, which is what I talked about here with our relay center to the cerebellum. We did not talk about this, so that's fine. The fourth ventricle we actually talked about a couple lectures ago, and so this would be the space that would be filled with cerebral spinal fluid. So hopefully that makes that a little bit more clear. I also realized that I had another little image here. When I was talking about decussation or crossing over, this is going to be crossing over of our motor neurons or motor fibers as they come through the pyramids. So we can see coming out of the brain, we follow these purple fibers. From the cerebrum, we're going to send motor messages down and then out through the spinal cord. At this point in the medulla oblongata, what is coming from the left side of the brain crosses over to be on the right side of the body, and what's coming from the right side of the brain crosses over and becomes part of the, or sends messages to part of the left side of the brain. And so we can see that point where those fibers actually cross from one side to the other. That is our decussation point. And for our motor neurons, that's going to happen in the pyramids of the medulla oblongata. So hopefully that clarifies things a little bit. Let's move into our pons now. So I only have one slide on the pons, so pretty much everything we need to know about the pons is here. It is an anterior bulge, and that bulge is towards the rostral region, and it's rostral to the medulla. So we have sort of the medulla here, which is basically a straight tube almost. Anteriorly or rostrally, we get this bulge coming up, and then we have sort of a flattened back here. That region, so shown here in pink, is going to be the pons. It looks like a backwards P. That always helps me identify where the pons is. It is going to contain, or sort of some of its major features are going to be cerebellar peduncles. They are going to connect the cerebellum to the pons in the midbrain. So peduncles peduncle. There's that fourth ventricle again. So this is going to be the anterior side, and this is going to be, or this would be the rostral side, and this would be back towards the caudal side. So we have these peduncles on either side of that ventricle. Those are going to, again, connect the cerebellum to the pons in the midbrain. So sending information from region to region, we are going to have lots and lots of integration and connection between our different brain regions here. We also have a continuation of the reticular formation. It does look different here in the pons than it did in the medulla oblongata, so it does change. But remember that the reticular formation is going to be that loose network of nuclei, and it does continue from the medulla through the pons and then even up into the midbrain. So it connects all of these different regions. Here, the reticular formation is going to be involved in sleep, respiration, and posture. So maybe different regions are going to assist in different things or different activities. We do still have the medial lemniscus. So again, it changes how it looks. Remember, in the medulla, they sort of looked like these side-by-side regions, sort of like that. Here, they are changing their trajectory, and they've sort of spread out a little bit and changed orientation, but they're still there. And so those are going to be our ascending sensory tracts. We also do have the presence of descending motor tracts, and so an example of that would be this tectospinal tract shown here. That's going to be a motor tract that's moving out of the body. And so, sorry, got distracted. There's weird noises upstairs. And so we also do have cranial nerves here, and so we can see those sort of coming off either side. So I don't really have a ton to say about the pons, aside from pointing out a few of those. We are just continuing these tracks for movement of sensory information to our higher brain regions, as well as the descending tracks to send information from those higher brain regions out to the rest of the body. And we do have some different features here that allow us to connect the cerebrum to the cerebellum, as well as the midbrain and the pons to the cerebellum. So again, we're just keeping all of our pieces integrated and connected to each other using these sort of brain stem or these hindbrain regions. So rather than move up into the midbrain now, we're going to move into the cerebellum. So the cerebellum is going to be the largest region of the hindbrain. It is the most notable in terms of structure. And so we do have a few slides on this brain region here. It is divided into hemispheres. So we have sorry, my laser pointer isn't working. There we go. So we have sort of two different regions, and like I said, this always reminds me of a walnut or something like that. Our two different hemispheres are connected by what's called vermis. So you can see this line of ridges or gyri and sulci. That's going to be the vermis that connects. It's basically a bridge that connects the right and left cerebral hemispheres to each other. We have three lobes. So we have an anterior lobe, a posterior lobe, and then the third lobe is the flocculonodular lobe. That lobe you can't see from an exterior view, and actually is sort of tucked up in here. It's a very small lobe, and so it's not evident really easily from just looking at external anatomy. Sort of like our cerebrum, we do have sulci and gyri here. This time we call them folia. Of course, we have to change the name. These are transverse ridges. So we can see that these are very sort of long and transverse. They go off to the sides. And they are separated by fairly shallow sulci. There is a thin cortex of gray matter, which we can see when we look at a cross section. So we can see this gray matter on the outside. Internally or deep to that gray matter, we do have white matter. This white matter is arranged in a orientation that looks like a tree, and so we call this the arbor vitae, or the tree of life. So it has sort of this fern-like branching, and again, we have a very distinct appearance here. All of the input and all of the output to the brain does also come through the cerebellum for processing. It contains about 60% of the surface area of the entire brain. So all of these ridges and folding give it quite a bit of surface area, and it also contains about 50% of the neurons in total that make up the brain. So the cerebellum is only 10% of the brain by volume, but it has a huge amount of surface area and a huge number and density of neurons. I also wanted to point out just our relationship of the cerebellum to some of our other structures. So here's our medulla oblongata, and then here's that pons, and then up here we have the midbrain. So the cerebellum is going to be caudal to our brainstem, comes off the back basically. It also is tucked sort of underneath or inferior to the cerebrum. Here is that fourth ventricle, so that's sort of that triangular shaped one, where above that we're going to have the cerebral aqueduct, and then heading down off of that we're going to go into the spinal cord, and we'll have the central canal. A few more little points about the cerebellum here. So the cerebellum is going to have all of its fibers being ipsilateral, which means that they serve the same side of the body. Fibers on the right side of the cerebellum are going to go to the right side of the body, and fibers from the left side of the cerebellum are going to go to the left side of the body. So they do not cross over. They are not contralateral here. They are ipsilateral. We have 3 fiber tracts that connect the cerebellum to the brainstem. And so these fiber tracts are going to be really thick bundles that are carrying signals both to and from. We are going to get the superior cerebral peduncles, which is this little region right here. That is going to connect this to the midbrain. So this would be the connection between the cerebellum and the midbrain. The middle cerebral peduncles, which is this one, is going to connect the pons to the cerebellum. And then inferior cerebral peduncles, so this sort of block right here, that bundle of nerve fibers is going to connect the medulla oblongata to the cerebellum. Okay, so now we're going to just quickly go through the functions of the cerebellum, and I'm going to go to the next slide. So we have a lot of major functions associated with the cerebellum. Sort of the big one is going to be to aid in motor coordination. So when we make a decision to send a motor signal to a part of the body, so let's just say we're going to move our arm, that information coming from the cerebrum can be very coarse and uncontrolled. The cerebellum is going to take that motor output information and it's going to coordinate it with what's going on in the environment around us and make sure that we don't just randomly flail and flail so hard that we hit a window and break it, right? So we are going to control and make sure that our motor movements are appropriate for the current situation. So the cerebellum is going to be really important in sort of making our movements fluid as well as appropriate. We also are going to use the cerebellum for evaluation of sensory input. Jeez. I'm really struggling this morning. Sorry, guys. Okay. Just monitoring our muscle movements. And I'm just making a list of different sort of functions of the cerebellum. So you can touch and feel, you can reach your hand into your pocket and you can touch and feel different surfaces in your pocket and know what you're touching, like keys versus chapstick versus a receipt. That would be comparison of our tactile stimuli. Spatial perception, and also appropriate timing of actions and when to do things. So this is just a short list of what the cerebellum does. So the functions of the cerebellum. I want to go through how cerebral processing works, so I'm going to delete these now. Let's talk about how the cerebellum actually processes things. So the first thing it does is it receives impulses from the cerebral cortex, which tell us that we're going to voluntarily move a muscle. So you're going to take a step or you're going to kick a ball. That information is coming from the cerebral cortex and it goes to the cerebellum for processing. We also at the same time receive signals from what are called proprioceptors throughout the body. So those are pressure receptors, usually located or associated with our joints. That tells us where we are in space. So are we sitting? Are we standing? Are we moving? Are we moving quickly? Are we moving slowly? What's around us? Because we're also going to use visual input and equilibrium information. We're going to integrate all of that, right? So we basically have 3 different sources of sensory input here coming from our ears, which is going to tell us our orientation, standing, sitting, spinning around. Proprioceptors, which tell us basically if we're moving or what joints are doing what right now. And then visual information, which tells us what our environment is, what's around us, do we need to be careful. So we have motor impulses, for a voluntary muscle contraction that wants to occur. We are now sort of scanning our environment and figuring out what position we are in, what we're doing, and what we're surrounded by. The cerebellar cortex is then going to calculate the best way to coordinate this muscle movement. So am I standing in a line of people? If I move my foot right now, am I going to kick someone, right? Your cerebellar cortex is going to calculate how to carry out that muscle contraction without causing a problem. And then the last thing it does is it sends a blueprint of that now coordinated movement to the motor cortex and then to the brain stem so that we can actually carry out that information. So the cerebellum is essentially taking all of these different sources of information and putting them together to make sure that whatever we're doing it safely and in a way that won't hurt us, won't hurt others, and that will actually carry out the process that we wanted to carry out in the first place. Okay, so I am going to stop here. The next little recorded lecture is going to be quite short. I'm just going to cover a few, maybe 2 slides, keep it short and succinct, and to wrap up this, what will be a short week because this will be Easter week. So I will talk to you in a few minutes.